Hello, everyone. Let's start this plenary session, and it's great to see so many of you here. My name is Mitka Lenasi, and I'll be co-chairing this session with Mario Gimona. And first, let's start with our keynote speaker. It is an immense pleasure to invite Dr. Alissa Weaver to the stage. So Alissa is contributed importantly to many key fundamental findings in the EV field. And she's especially interested in the EV's role in cell motility and how this also affects cancer or tumor metastasis. And on a personal note, Alissa was actually chairing the session where I gave my first talk at an ISEV meeting. So I think this is a nice circle. And I hope you will enjoy the talk. And please, Alissa, we're happy to hear about your data.
Thank you so much. So it's really an honor to be able to give a plenary talk at ISEV. And so I just want to tell you a bit about our journey. So we first started becoming interested in exosomes because we were interested in cell migration, and along the way figured out that exosomes were a key part of that. So I've always been interested in this process of cancer metastasis, how cancer cells spread from one part of the body to another. This involves cancer cells migrating and invading out of primary tumors and traveling to distant sites, migrating back out of the bloodstream or the lymphatics, and colonizing distant sites. And so in order for cells to migrate, they make a variety of actin-based structures. I actually did my postdoc on the actin cytoskeleton, and the upper right panel for you is a migrating breast cancer cell. The red is actin, the green is a cellular adhesion molecule, and you can see leading edge lamellipodia at the front of the cell, green adhesions, actin bundles. And then at the bottom, you can see a breast cancer cell that's been plated on a bed of fluorescent extracellular matrix. And those cells are making these structures we call invatopodia, which are actin-based protrusions that co-localize with sites of extracellular matrix degradation that you can see as the black holes where the matrix has been digested and then endocytosed. And actually, Mario and I were both in the invatopodia field together for a long time. And one of the things that we realized early on in my laboratory is that these invatopodia are really hot spots for secretion of proteinases. And so what you can see in this movie, maybe look at the merge of the red and the green, is that the actin spots show up first. That's the invatopodia initiating. And then a green proteinase called MT1-MMP, which is this really key transmembrane proteinase that drives digestion and invatopodia function, is secreted over time at these invatopodia. And we were trying to understand how that happened. And this led us to exosomes, basically. So Philippe Chavrier's laboratory showed that MT1-MMP was probably coming from late endosomes. And then we looked to see whether some exosome markers, CD63, were co-localizing at sites of degradation of invatopodia. Indeed, they are. You can see that in that image with the blue, where the dark holes is the degraded matrix. We also purified small extracellular vesicles and found that MT1-MMP and other proteinases were actually carried by the vesicles along with other exosome and small EV markers. And so this was our first foray into the exosome field, and we published a paper showing that actually multivesicular bodies are docked at invatopodia and secrete proteinases there, and that exosome secretion is important for both invatopodia activity, but also the formation. There's a positive feedback cycle. I also want to point out that large extracellular vesicles also carry proteinases. So the shed microvesicles image, that's from work from Chrislyn D'Souza-Shori's laboratory, where they showed that gelatinases are carried on large vesicles. And so This is a property that multiple kinds of vesicles have. But we've mostly focused on the exosomes, and this is just a number of our papers where we showed that Invatopodia depend on exosomes, that this is regulated by branched actin assembly, in particular this marker cortactin that controls actin assembly, endosome dynamics, and multivesicular body docking. So we've also looked at the role of exosome secretion in cell migration, and some of that came from our cortactin work because we knew that cortactin had knockdown cells had migration defects, and we thought it might relate to exosomes. But to really look at this definitively, we inhibited canonical exosome secretion regulators. So in this case, RAB27A and synaptotagmin 7, which inhibit respectively docking and fusion of multivesicular bodies at the plasma membrane. And so what you're looking at here is cells that are migrating in a metastatic chorioallantoic membrane from a chick embryo. So we inject fluorescently labeled cancer cells, and then we image them at the metastatic site. And if you look at the individual cancer cells migrating where it says SC, that's scrambled control, I hope you can see that the cells are migrating fast and directionally. Whereas for the exosome-inhibited cells, they'll start to migrate in one direction, but then they rapidly lose their polarization, and they don't really go anywhere. This is perhaps better appreciated when we've overlaid some of the migration tracks from the movies from which we do quantitations, and you can see that the control cells really migrate directionally, and the exosome-inhibited cells really do not. And the plots below, that's called a Windrose plot, where you put all the tracks around the clock, and in the very middle are the exosome-inhibited tracks. And so if you look at the zoom out, there's really no directionality to the migration of those cells. And so we published a number of papers. Bongwon Sung, who is here at the meeting and presented a poster over the past couple of days, did all this work. He's a research associate professor in my lab. And what we found through these many manuscripts is that cancer cell chemotaxis, cancer cell speed, and nascent adhesion formation all depend on exosome secretion. And we used a variety of genetic inhibition and exosome add-back experiments to do that. And I always like to point out that, even though we work in cancer cells, we like to see that our results relate to other cell types, other contexts. And Carol Perrant's lab has done some very important work in this area as well, looking at neutrophil and Dictyostelium amoeba chemotaxis. And so the image that's shown over there is from one of her papers where they inhibited RAB27A and neutral sphingomyelinase and found that neutrophil chemotaxis towards the bacterial peptide F-metlu-Phe is compromised and depends on exosomes. So we think it's a universal property. Okay, so now I want to talk a little bit about some tools that we've made for the field. So if you study migration, as you've already seen, a lot of it is about trying to understand the cytoskeletal structures that are formed and taking movies and trying to observe how one thing relates to another. And so we wanted to have the ability to see where exosomes are secreted and how does that relate to cytoskeletal structure formation in migrating cells. And so to do this, we took an approach which you've probably already seen some of at this meeting, if you attended Guillaume Van Niel's talk, of hooking a pH-sensitive GFP to an exosome marker, in this case CD63. This is an approach that neuroscientists have used for a long time to track synaptic vesicle fusion. Basically, the fluorine GFP is quenched in the acidic environment of the multivesicular body, and then it fluoresces at neutral pH, which is what happens when an MVB fuses with the plasma membrane. Okay, so this was really early days, and first we did some movies of migrating cells, and we were fascinated to note that there were these apparent exosome trails that were being left behind cells. And we don't see plasma membrane markers being left behind. It's really just the CD63 that's being left behind. So we then used total internal reflection fluorescence microscopy to be able to track the relationship between the exosome secretion and new adhesion formation, because we had identified this defect in cell speed that cells have. And so if you look at the arrows, you should be able to see a green flash that quickly precedes a red adhesion formation in these movies. It's very quick, and these movies are every 30 seconds a frame. But we pretty much saw that almost always there was a green flash before a red adhesion formed. So, as I mentioned, Guillaume Van Niel, Michael Pegtel, and Frederick Verwedge have also made a CD63 fluorine. They've placed the fluorine a few amino acids away from where we did. And we and others have used this to track MVB fusion with the plasma membrane and exosome secretion. But there was a drawback to the original construct that we made. So we found that it was pretty dim, which means then that you have to increase light, and there's the possibility of phototoxicity. We also like to stably express fluorescent constructs in cells and then flow sort cells to get the appropriate level of fluorescence, so it's not too little, not too much. And we weren't able to do that. So we looked in the literature, and we found that, yes, indeed, fluorine can be unstable. And there was a group that was looking at this in bacteria and made a point mutation, M153R, that stabilized the fluorine for them. So we tried that in our construct, and indeed it's a much brighter probe that's stable. Now we can use lentivirus to make stable expressing cells, and you can see these very beautiful exosome trails that are left behind the migrating cell. So the M-Cherry-Cax marker in this case is a plasma membrane marker. You can also see it in endosomes because, of course, the plasma membrane gets endocytosed, but what I hope you'll see is that the plasma membrane marker is not being left behind the cell, just as CD63 is. And this is just showing at the front of the cell and the back of the migrating cell. So we published this construct in 2020. It's available on AdGene. We wanted to really definitively show that it's an exosome marker, so we did many studies, one of which is here. We can see colocalization with other exosome markers like ALEX in these deposits that are outside the cell. We also did correlative light electron microscopy. And so I hope what you can appreciate on the bottom is the zooms from some of this CLEM work. The middle bottom panel, you can see that the green spots really correlate with small vesicles, whereas a larger vesicle is not green. And then to the right, we can also see some structures that look like microsomes. And this is just larger, so you can see those structures a little bit better. So we've used this construct to look at cell migration, and it's amazing to see how these trails that are put down, the cells just love to migrate on them. And I'm not showing the quantitations that we've done here, but I hope you can appreciate from the movies how the cells find those tracks and really like to migrate along them. And some of what we showed in our earlier paper was that actually there's extracellular matrix that's bound to integrins on the exosomes that drives the nascent adhesion formation and promotes the cell speed and the tracking of the cells. So we think that that's a lot of what's happening there. We also see this in 3D, that the cells are following these tracks. So one of the things that we noticed when we were doing our movies is that we could see these other structures called phylopodia, and that they were associated with exosome secretion. So in this current movie, maybe if you look at the red-green merge, it will be easiest to see. If you look at the arrowhead, that's going to be the first exosome secretion, and then rapidly thereafter, the arrow is showing the phylopodia that protrude. So we noticed this association. This now is a dual label construct that we made also available on AdGene, where we've got the pH-sensitive GFP that shows you the exocytosis, but we can also track the MVB within the cell because we put a pH-insensitive M scarlet at the C-terminus with a linker in CD63. So it's a very useful construct. So based on those movies, we hypothesized that exosomes might control Filopodia formation. And indeed they do, and I'm going to show you the data for that now. So we took our approach that you may recognize already by now of the genetically inhibiting exosome secretion, in this case, knocking down either Rab27A to inhibit multivesicular body docking, or HRS, escort 0 protein to inhibit exosome biogenesis. And we see a selective decrease in the small extracellular vesicles that are secreted from cells, no effect on the large extracellular vesicles. And in the images, I hope you can appreciate that our control SH-Z melanoma cells are making numerous Filopodia, those slender protrusions from the cells, and the knockdown cells are very smooth with no or very few Filopodia, and that's quantitated below in D. And then we also like to really show that the exosomes have a responsible factor by adding back purified vesicles. And in this case, we've added back both purified large extracellular vesicles. This is purified by a 10,000 G ultracentrifugation, or purified small extracellular vesicles. And we typically purify those by cushion density gradient fractionation. And you can see that the small EVs, but not the large EVs, can rescue the Filopodia formation. So Filopodia are very interesting structures. So they direct cell migration. They've been associated with cancer metastasis, angiogenesis. One of the things that they do is they promote synapse formation by neurons. So it's a very clear function for Filopodia in tissues. And so we wanted to look at that as well, and collaborated with Donna Webb's laboratory and a very talented graduate student, Meekan Patel, who isolated rat embryo, both cortical neurons and hippocampal neurons, and would transfect those neurons with shRNAs to affect exosome secretion. In this case, we used Rab27B because neurons don't really express Rab27A, and it worked. And we also used HRS shRNA. And another thing you need to know is that for these embryonic neurons, they sort of develop over time, and the maximum time for Filopodia formation is day in vitro 6, so that's when we quantitate that, and then they turn into spines and synapses, maximum around day 12, so that's when we quantitate the spines and synapses. And basically, the exosome-inhibited neurons have a decrease in Filopodia at day 6, and spines and synapses at day 12. And Meekan also was able to develop a way to purify the small extracellular vesicles from these primary neurons in culture and add them back and rescue the Filopodia formation. And that's shown on the bottom in I and J. You can see the decrease in Filopodia and then the increase. I know the writing is really small, but basically that's the knockdown and then rescue by adding back purified extracellular vesicles, either for B in one graph or HRS in the other graph. Okay, so I've showed you that exosomes can control Filopodia formation, but actually, what are the cargoes that are doing this? So we didn't know. So we do a lot of proteomics in the laboratory. This approach was actually very old school proteomics, where Daisuke Hoshino, a postdoc in the lab at the time, purified large extracellular vesicles and small extracellular vesicles, ran them on a gel stained with colloidal Kumasi so that we could then cut differentially dark bands or differentially expressed bands out and send them for mass spectrometry. And he did that for 4 different bands. And one of the interesting proteins he found is called endoglin. And From a Western blot in the middle, you can probably see that endoglin is well expressed in small extracellular vesicles. And then Nanyang Hong did a transient knockdown of endoglin and found that indeed in our cancer cells, we found a decrease in filopodia formation in the endoglin knockdown cells. So it seemed like a good candidate to pursue as an exosome cargo that could be driving this function. So what is endoglin? So endoglin is a TGF-beta family co-receptor. It modulates TGF-beta type 2 signaling, so it binds to signaling TGF-beta receptors like ALK1 and ALK5 to TGF-beta ligands. And it's expressed highly in endothelial cells, and in fact, it's a disease gene for human hereditary telangiectasia. So filopodia are very important for angiogenesis, so this was interesting. Also interesting is endoglin is upregulated in a number of cancers. So remember, that's how we got to endoglin, was from cancer cell EVs. So we did our usual knockdown rescue approach. Indeed, we can rescue filopodia defects of endoglin knockdown cancer cells by adding back purified small extracellular vesicles, not with large extracellular vesicles, and also not with extracellular vesicles purified from endoglin knockdown cells. So there really is a specificity to the cargo content coming from the endoglin expressing versus knockdown cells. And we could do the same thing with HRS knockdown cells. The control EVs rescue the filopodia defect, whereas the endoglin knockdown ones do not. Also, I want to mention that filopodia are related to cancer cell migration, invasion, metastasis, and so we did our chorioallantoic membrane assay again, in this case looking at metastatic colony formation, where we inject green cancer cells, GFP-expressing cancer cells into the vasculature of the chick embryo. 4 days later, we harvest the chorioallantoic membrane and we assess metastatic colony formation. The endoglin knockdown cells formed few and large colonies. The larger colonies are often indicative of a defect in migration away, so they can still proliferate at those metastatic colony sites, but they can't migrate away. And if we co-injected purified extracellular vesicles with the endoglin knockdown cells, that rescued the metastatic colony number and size defect. And I mentioned those large colonies might indicate a migration defect. Indeed, if we look at spheroid migration in type 1 collagen gels, we see that endoglin knockdown fibrosarcoma cancer cells have a defect in migration in this assay, which in this case we didn't add exosomes back, but we can rescue it by re-expressing endoglin in the cancer cells. Okay, but how is endoglin really promoting filopodia formation? Well, we thought it probably is due to endoglin because it's a co-receptor carrying some of its binding partners with it, and then those must be interacting with the recipient cells and inducing filopodia formation. But we looked at the obvious candidates and we really didn't see any difference. So the endoglin knockdown EVs still had TGF-beta, still had integrin beta-1, still had ALK1, which is a TGF-beta receptor. And furthermore, when we tried to rescue filopodia defects of endoglin knockdown cells with TGF-beta or BMP9, or plate the cells on fibronectin, none of those things rescued the filopodia defect. So there was clearly some other cargo that we were unaware of that was on those EVs and regulated by endoglin. So we carried out another proteomic screen. In this case, we use iTrack, where you use isobaric tags, label in equal amounts of proteins, mix a protein together, and then you get an absolute ratio of protein enrichment. And really interestingly, besides endoglin, the only cargo that we saw that was decreased across multiple samples in the endoglin knockdown EVs was an ECM protein called THSD7A. So that stands for thrombospondin domain 7A. So it's a transmembrane molecule that has extracellular matrix domains on the extracellular part. And interestingly, very few papers about it, but it had been identified as a regulator of filopodia in both endothelial cells and neurons. Both cells really depend on filopodia. And it has both transmembrane and soluble forms, and we think that probably a lot of the soluble form that has been reported is actually carried by extracellular vesicles. It's also a disease gene in autoimmune kidney disease. So, interestingly, this is now the second disease gene that we found in our screens. And there was an interesting talk earlier in the meeting about actually these autoantibodies to THSD7A that cause membranous glomerulonephritis in patients, and that can induce exofer formation from kidney podocytes. But in this case, we're seeing it on the small extracellular vesicles that are getting secreted. Okay, so is it really regulated by endoglin on the small EVs? Indeed it is. So in panels A and B, I hope you can appreciate that endoglin knockdown leads to a decrease in THSD7A in small extracellular vesicles. Also, THSD7A is expressed on small extracellular vesicles from neurons. That's in panel C. So this is very important because I've just shown you a lot of data about endoglin and filopodia formation, but we always had a sort of a problem pursuing that cargo because neurons don't actually express endoglin. So we really wanted a universal exosome cargo that could explain how we can see the same phenotype on filopodia in both cancer cells and neurons. And we think THSD7A is that universal cargo. So I did mention THSD7A has been reported to promote filopodia formation in the past, and indeed it does in our hands. So if we express an M-scarlet version of THSD7A in our cancer cells, we see it at the tips of filopodia, and we also see it in these extracellular deposits that look very much like our CD63 positive exosome deposits, and I'm not showing it here, but we can see colocalization with CD63 and THSD7A outside the cell. If we quantitate filopodia formation in the cells expressing the THSD7A construct, there are more filopodia. And also, if we knock down THSD7A, we see fewer filopodia in our cancer cells. And again, we like to rescue phenotypes. Here we can come back with just a purified recombinant version of THSD7A, which conveniently was commercially available, and find that we can rescue our endoglin knockdown cells just by plating those cells on THSD7A more filopodia. And indeed, we can do the same thing with the neurons. So we can express THSD7A in the neurons. We see it at the tips of filopodia in the neurons. And we can take our HRS knockdown neurons, plate them on THSD7A, and find that it fully rescues the filopodia defects of exosome-inhibited neurons. So these and a lot of other data are available either on bioRxiv or as a reviewed preprint in eLife. But basically, our model is that exosomes containing THSD7A are secreted from cells. They're interacting with the cell surface with a receptor that we don't know. So we don't know what THSD7A is binding to on the cell surface to induce filopodia formation. That's a study for the future. But we have done some studies with CDC42 inhibitors, and we think that the signaling is going through CDC42 in both cancer cells and neurons to induce filopodia formation. In cancer cells, the presence of THSD7A on exosomes is regulated by endoglin, but we don't know how that happens in neurons. So just to summarize, I covered a lot of ground here, but I hope I've convince you that exosomes are critical mediators of cell migration. They affect multiple cytoskeletal structures, including adhesions and filopodia. They affect both cell speed and directional sensing. I didn't really show you the data for fibronectin being carried via exosomal integrins, but that's in a couple of our papers from 2015 and 2017, driving cell adhesion formation and cell speed. I showed you the data for THSD7A, driving filopodia formation. And using our reporters, we really see that cells are pathfinding and have leader-follower behavior along exosome trails. So I think there are many open questions. This is really a fascinating area. So why are exosomes so essential for migratory behaviors? So we've identified some of the molecules, but there are many more that we've not identified. So for example, we don't know in cancer cells why exosomes are required for directional sensing and chemotaxis. There's a ton to be discovered there. There's a lot more to be discovered both for the cargo side, but also the cell receptor side of how this is working, how the key cargoes are regulated, and then how does this play into disease pathogenesis. I mentioned we keep identifying these disease genes as cargoes in extracellular vesicles, but we don't really know exactly how that plays into the pathogenesis. So I'd like to acknowledge the many people in my laboratory that contributed to this. This is actually a picture in my backyard from a lab party. Bangwon Sung has really done almost all of the migration work in my laboratory and really driven this. The filopodia work was started by Daisuke Hoshino on the right there. He has his own laboratory in Japan, and then carried forward by Caitlin McCarthy on the cancer cell side and Meekin Patel on the neuron side, as well as some help from Nanyang Hong, a former graduate student in the laboratory. I would like to point out also we have a Center for Extracellular Vesicle Research at Vanderbilt, so check out our website sometime, and I'm happy to take any questions. Thanks for your attention.
So thank you for this fantastic talk, and I'm sure there'll be several questions, and we have time for them. So come up. If you have any questions, please go ahead here.
Yeah. Oh, thank you, Alissa, for this wonderful talk. So I have one question and one remark. So the question is about when you rescue your effect with the small EV versus large EV on filopodia formation, right? And you do that with small EV that have been prepared by ultracentrifugation. Do you see kind of loss of activity in the pro-migratory effect, in the sense that, for example, for us, when we look at the pro-migratory activity of EVs, we found that if we prepare them by ultracentrifugation, they lose up to 90% of their activity. So in a way, we have to give 10 times more than if we just have the conditioned medium. You understand my question?
Yeah, I understand your question. I would not say that we've done like a super comparative across like that, but one thing that we do for the purification and our add back that I can speak to that. So first of all, we generally use a cushion density gradient, and the way that works is you're not forming a pellet. So you're taking the conditioned media, ultracentrifuging it on top of an iodixonal cushion, so you sort of get a layer of the extracellular vesicles, and then you take that layer and then you float it up in a density gradient. So at the end, yes, you form a pellet when you're washing everything, but it kind of minimizes the aggregation and the contaminants. And we generally find that the EVs are functional. We haven't done a super rigorous cross-comparison with size exclusion or other methods. But I will say that one thing we do is we calculate how many vesicles are secreted per cell per hour for our preps, and when we do add backs, we try to do it in a dose concentration around what we would calculate to be the amount of vesicles that are secreted from the cells. So based on that secretion rate and how many hours the assay is going for and how many cells we have in our dish, we will add back that number of purified extracellular vesicles. And we're generally in kind of the right ballpark for our dose concentration curves. We might need to use like twice as much as we would calculate to be secreted from the cells in our, to get a full rescue of our phenotype. But I think since you're probably losing half in a density gradient anyway, it's pretty good.
Okay, so it's pretty good.
So go ahead. On the left.
Outstanding talk. So, as you may know, the ARP23 complex and the WAVE and WASP protein are drivers of Filopodia cell migration invasion. I was surprised not to see these in your profiling, proteomic profiling of exosomes. So, are you saying that these they are not part of the cargo and they form Filopodia independently of exosome?
You're talking about the Invadopodia that we were showing early on?
No, I'm talking about the general process of Filopodia formation, which is driven by the ARP23 complex and the WASP and WAVE proteins.
Well, there's always been a debate about branched actin assembly versus unbranched actin assembly for Filopodia formation. But what we're saying is we're not really focused on the nuts and bolts of the actin part of this process. We're focusing on the extracellular signal that's coming in to induce the actin assembly for this project. So, my postdoc was in the actin field, and I very much understand like the heavy actin focus of how all these proteins induce these actin structures. But one of the things that intrigued me was there actually had not been that much focus on what are the extracellular signals that are coming from the outside to signal that this structure initiates in the first place. And so what we're focusing on here is really the outside signaling rather than the inside. Yeah.
Thanks for your presentation. My question is, do you have any idea or hypothesis about how the endocrine split protein can selectively packaging the THSD7 into the intradermal vesicle and participate in MVP biogenesis and small UV secretion?
Yeah, this is definitely like a topic for lots of future investigation. I can tell you what we've done. So we have visualized THSD7A and CD63 in control and endoglin knockdown cells, and it's really interesting because whereas we would think that there would not be THSD7A in the MVBs in the endoglin knockdown cells, there's more there. And so we think that actually those MVBs are maybe not getting secreted. But we're not completely sure. Like, we need to do more high resolution studies to really rule in or out that there's a biogenesis problem in the endoglin knockdown, but so far our data would suggest that actually the MVBs containing THSD7A are just not being secreted in the endoglin knockdown cells, and we really don't understand how that's happening. Thanks.
We'll move back to the left.
Thanks, Alissa. So I'm just wondering, with your THD protein, when you put the recombinant protein, I'm assuming that is a soluble form, but when you have your EVs, this is a membrane protein. I'm just wondering, can you compare between that in terms of the functional state?
Yeah, I mean, we've done both exosome, coding and matrix coding, and we generally kind of do them the same way, and they both rescue. We just code it like any other matrix, just like you would fibronectin, overnight, 4 degrees, dose concentration, and yeah. I know it's a transmembrane protein and we buy it recombinant, but we think it's the extracellular domain that's the active player there. Yeah.
Thank you.
Go ahead, please.
So, Alissa, your
the
work
right.
beautifully suggests the laying down tracks and following those tracks. And I'm wondering with your live cell microscopy, if you might be able to dissect that this may be a two-step process, that maybe the thrombospondin and endoglin may be related to the first stage, and then integrins may be the second stage, or have you seen evidence for differences in those filopodia at those different stages?
We have not done the kind of microscopy to really distinguish between, like, are the filopodia coming first and then the adhesions coming next. It's a pretty fast process too, so we would have to take much faster movies to do that. It certainly could be done. And filopodia themselves are sort of adhesive structures, so it's sort of, everything's happening very fast. I will say our ability just to capture the exosome secretion before the cytoskeletal structure formation, you know, it's all happening within, you know, 30 seconds to a minute. So it's a pretty fast process. So we would need to...
Maybe some additional knockdown and add-back experiments.
Well, or maybe like a 3-color movies to really like dig down on the timing of that. Yeah.
Thank you.
But I will say, you know, I mentioned it briefly, we cannot rescue filopodia formation by plating cells on fibronectin, but we can rescue cell speed, and we know that those are separate cargoes. And so we think that those are really distinct aspects, so that there are multiple exosome cargoes that are regulating specific distinct parts of the migration process. Yeah.
Will you move to the left and back to the right?
Yeah, sorry,
Oh,
Pascal. Thank you, Alissa. A very beautiful talk. I have a question. You really focus on exosomes, and we know that also these cancer cells make microvesicles. If they're migrating, they have to be polarized. Do you know how the microvesicles, what their content is as opposed to the exosomes? And my guess is that the microvesicles are not secreted in a polarized fashion, and maybe the exosomes are. Have you looked at something like this?
We have not looked at microvesicle secretion. We do use it in our add-back assays and have not found a lot of activity in the, you know, different assays that we're looking at. I have to say we haven't spent a lot of time sort of trying to make microvesicle markers or manipulate microvesicles to follow that activity. But some of the exosome cargoes that we see that are driving migration, they really are enriched on the small EVs compared to the large EVs. So I mentioned extracellular matrix and integrins. We've done comparative proteomics, and we see heavy enrichment of integrins in small extracellular vesicles compared to large ones, doing an equal protein comparative proteomics. Same thing for the THSD7A, we really see it on small extracellular vesicles. So for the cytoskeletal reorganization, for the activities and cargoes that we're tracking, it really does seem to be the small EVs. But no, we haven't really looked at microvesicles and whether that's involved in polarization as well. Yeah.
So 2 short questions.
Yeah, okay, so it's not a question, it's a remark. So maybe a source of inspiration for the molecular mechanistics behind your crosstalk between filopodia and exosome tracks. So looking at AML stroma communication, we found that if we knock down synthenin in the stroma, we have more aggressive AML, and this is because synthenin controls the balance. So synthenin binds directly to endoglin and controls the secretion of endoglin in exosomes. And if you have an imbalance, because a loss of function of synthetin, this puts a vicious communication circle between the stromal cells and the AML cells. And this is all about endoglin on EVs. And behind, this is also an imbalance in protein translation in the tumor cells. So maybe for the mechanistics aspect, I think there is some parallel over there that might be inspiring. It's published in EMBO Molecular Medicine 2023.
Yeah, endoglin's a very interesting molecule. It's also a mesenchymal stem cell marker that people use, and I think there's a lot to be learned there.
Just one short question.
Yeah.
Great talk, Alissa. So
Thank
for
you.
a long time, exosomes and EVs are being viewed as text messages between cells. The belief that they can travel 1,000 miles away to hit another cell. But based on your finding, if lots of these exosomes and EVs are being left on the trail, these text messages are being grounded. So do you think this is complementary or in some sort of way in contrary to the belief that exosomes can be used as cell-cell communications, or they're more about movement, really?
Well, I think it's complementary. I mean, clearly they're cell-cell communication devices, but we also see that exosomes are changing the microenvironment and directly interacting with cells and on the outside and changing their phenotype. So I think they have multiple activities. And one of the things specifically for directional cell migration and chemotaxis, there's always been a mystery about how cells could sense a chemokine gradient from the front to the back of a cell, because it's such a shallow difference, from the length of the cell in the gradient. And I think that having these secondary mediators, which exosomes are, they get, Carol Perrant showed that in response to the soluble mediator, you get the exosomes that then are a stable trail, that it's a way to enhance the communication and support like a positive feedback cycle for the directional sensing of cells as they're navigating their way through the environment. So exosomes and extracellular vesicles in general, they have these unique properties, right? So they can carry unique cargoes, they can stably interact with the environment in a way that's a little bit different from soluble mediators. And so I think that this is the second way the vesicles work, is through direct modification of the environment that cells can interact with. Yeah.
Thank you so much for this lively discussion. And Alissa, thank you for joining us in Vienna.